Justen Whittall, and he is an associate professor at Santa Clara University. And he is going to be talking today to us about Metcalf Canyon Jewelflower.
Thank you, Nick and Jeanette, and especially to the Native Plant Society for bringing us all together as a community and providing a forum for us to exchange ideas like this. We've heard several talks already that touched on flower color, and I'm going to emphasize another example here. I'd like to quickly give you a background on this project. I started as a postdoc and was fortunate enough to be at UC Davis when Sharon Strauss was transitioning from the weedy radish to the native jewelflower. As a kid, growing up in the Santa Teresa Hills of southern Santa Clara County, I knew exactly where you could find some of these, and that information became immediately invaluable to that community at Davis, and I joined her on many a jewelflower hunt, which then spiraled into a grant proposal and a baseline biological data project for endangered species, the Metcalf Canyon Jewelflower. I've since met a lot of great people, especially local people here from Creekside Science, Sue's in the audience, and Christelle, as well as a lot of Native Plant Society volunteers who made this happen, so I thank them for that. Let me just quickly mention that Serpentine Endemics are obviously the best plants in the world. Although the columbine up there is very close to my heart, that's the Serpentine Seep Endemic Acknowledgements eximia. I'm going to talk to you primarily about the jewelflower on the far right, and this was one of my first finds as a kid, Dudleya Seychellii, taken from a picture here looking southwest across the Silicon Valley, formerly the Blossom Valley, and we hope to return it that way shortly. Metcalf Canyon Jewelflower, Streptanthus albidus, subspecies albidus, is a serpentine endemic. It's an annual herb. It's in the mustard family. It's listed endangered. But it's been historically rare as far as we know. This little slice of land here is Coyote Ridge. To the east is the Diablo Range. And it's primarily known from the northern half of the Coyote Ridge. This collection from Los Gatos and further south near Gilroy have been relocated and identified as Streptanthus glandulosus, a much more common taxon. I'll tell you where we're going today. That was the short background. I'd like to dive in and tell you a little bit how to tackle these flower colors, because it's clearly a problem, whether Brian was talking about in Delphinium and Tommy was just mentioning it in Claytonia. We can quantify that. We can ask whether the pollinators care about those colors. In fact, we can look for correlates with soil chemistry and eventually get to some molecular markers, which in this case don't seem to help very much. But alas, we use the biological data that we have acquired to try to make a change for this endangered species. So focusing on the flower colors here, this is known as the Streptanthus glandulosus complex, and in fact this is just a couple of the taxa that Mike Myers and his student in 2010 published in this very cryptic paper. It should look a little bit fuzzy to you. In fact, the taxonomy is sort of in flux, but he's used ITS into the chloroplast restriction sites to identify some very well differentiated geographic clades. You can see them in the north and central, eastern, and south, and in fact they're going to focus on this southern clade, which in and of itself contains several formally described taxa, like the Metcalf Canyon Jewelflower, the most beautiful jewelflower, the more common glandulosus, and this enigmatic taxon from the top of Mount Hamilton. We've now found it in a couple of other places. Heath Bartosh was reminding me that it's also Revaisanol. So flower color, although one of the most inviting things about angiosperms, and at the same time one of the most perplexing things in taxonomy, can be quantified, and you can quantify it in many ways, and my lab's taken several approaches to this, but in this case we thought we would just take a UV-vis spectrometer out to the field, and you can measure the percent reflectance on the y-axis and the wavelengths. Just to orient yourself, we can see between 400 and 700 nanometers roughly, and a white flower reflects most of the light between 400 and 700. Again, we're looking just at the sepals in this jewelflowers. These are these urn-shaped, showy sepals that make jewelflowers unique, especially in this group. In contrast, the most beautiful jewelflower, subspecies Paramoenis, absorbs a large amount of the wavelengths of light, around 550, and that's characteristic of anthocyanins, particularly in this case cyanidin, that we know from some HPLC work. And as the flowers get deeper purple here, you'll see more of the light is absorbed, or in this case less is reflected, and that nearly dark one absorbs the majority of the light. Most important also is that there's no UV reflectance, so what we see is largely what bees would see, but possibly in different colors. For example, the bee color vision hexagon, defined by Chidka in 1992, takes that spectral data and puts it into the neurological framework of a bee. What color would they see? Now the details of this is fascinating. I'm not going into much except that they connect these electrodes to the antenna of the bee, they shoot light at them and see if they get excited or not. Sometimes you can do it with just the antenna, the bee doesn't even have to be alive. There are a lot of complications with that being said. We can put these points on here and bees would see, we have the white one, the light purple one, and the dark purple one. I don't have the black one from Mount Hamilton up here. But in fact, the white ones do seem to be different from the other two, and the other two seem to be separating out from left to right. I don't want to go into too much detail here, except that one, this is defined based on honeybees, because that's what we know most of, and we'll find out later they are not the primary pollinators of this taxon. And as you get closer and closer to the center here, the flower color becomes indistinguishable from the background, assuming the background is green, which works for honeybees, maybe in your garden. Hopefully your garden is green, but not in most cases of the jewelflower. By the time it's flowering, these serpentine grasslands, the background is anything but green. So let's ask the pollinators if they care. First, we need to identify who the primary pollinators are, and we use digital camcorders, complements of the Santa Clara University Media Services Division. Not just one, but often several students will come out to the field with me here at Metcalf Motorcycle Park in southern Santa Clara County. We'll line up these digital camcorders on tripods and set up, in fact, many at a time. In this case, we had 10 running from sunrise to sunset. And the great thing about this is that the students who are always diligently taking notes are often less familiar with the differences between solitary bees, honeybees, bumblebees, and it's all recorded. Meanwhile, I sit back and take care of business. And after 65 hours of footage and a lot of popcorn movie watching, we found the primary pollinator. In fact, more than 99.5% of visits were done by this two-striped, on the back end, yellow-striped bumblebee, Bombus vosnesenskii. And if technology is working my way here, we might see one. That's Malik Road, if you're familiar with this area that connects the Metcalf Road to Kirby Canyon, and I'd like to just show you what those bumblebees, how they behave. They'll come in to the view here, and you will see them moving and working the flowers from the bottom to the top. They're primarily collecting pollen, although there is about a microliter or two of nectar at the base of the flower. A lot of these plants do quite well on road cuts, whether you're on Metcalf Road or the adjacent areas. We'll see disturbance becomes quite important for them. All right. So we know the primary pollinator is this bumblebee, but what I want to test is whether or not bumblebees care, whether you're white, pink, dark purple, or in this case, we found, my study was inhibited for several years because we didn't know of any population where we could do this without releasing genes of the wrong flower color into, for example, the endangered species population, which would be a bad idea. We found a mixed population right between Kirby Canyon and Metcalf Canyon where we set up these arrays of picked inflorescences in water picks to keep them fresh. And we again used digital video camera technology here, pointed down at these arrays, and there's a student and my technician, Aaron Tom, and we did this, in fact, some 6 times in order to get a significant study going. We had these hexagonal arrays arranged such that there were 6 inflorescences around the outside with the same number of flowers in each, and something in the center, and we had 3 with whites in the center and 3 with purples in the center, and that turns out to be important because bees tend to go to the closest flower unless they have a strong preference otherwise. So if we just look at who showed up to this experiment, you'll see the number of visits were by and large bumblebees, which wasn't surprising. We did get a couple of honeybees, and although we call these flies, the resolution at this point is that distance was difficult, so we're not 100% sure what those are, but there was over 200 visits recorded, and the large majority of them were from these bumblebees. Well, which colors do they prefer? There's clearly not a really strong preference for either of the two, but maybe we can take advantage of the hexagonal array to get more details. For example, when a pollinator comes into view for us, which we assume is a new visit to the array, did they go on that first visit to a white or a pink? Well, 21 times they went to a white and 25 times they went to a pink, because they're not substantially different. Again, here's the array, and let me tell you what happens when you get a transition from the center plant. The whole point of these hexagons with a center plant is that a pollinator at this point has equidistance to go to any of the other 6 periphery flowers, and at the same time it's the same physical distance. So for example, if there's white in the center, there are 3 whites to choose from and 3 pinks to choose from at equal distances. So if the center was white, for example, we can count the number of transitions that they make from white to pink, or do they stay white on white? And in fact, they were exactly the same. Although their numbers are relatively small, the point here is that they either stay the same, or if the center of the hexagonal array is pink, in fact, they prefer to go to white than to go back to pink. The probability that they are choosing to stay the same color every time is very low. They either choose them equally, or in fact, sometimes they even prefer a change. These numbers are also quite low because we're limiting ourselves to just transitions where they started at a center plant. We can take advantage of one more aspect of the hexagonal array here. I'll challenge your mind here with a little bit of probability. That is, for example, if we look at these peripheral flowers and a pollinator lands on white, with white in the center, would you agree that they have 2 pinks to choose from that's the same distance roughly to the white? That is, there should be 2 pink visits for every white, assuming there's no preference. And magically, if we're calling it the same versus different, there were some 7 or 8, 9 visits to the same and some 22 visits to different. That was if you had white in the center. And as if they knew the probabilities to expect, there were 7 pinks and 14 pinks if they went to the different. 7 for the same, 14 for the different. We're getting stronger and stronger evidence for bees telling us they don't care. It's not surprising, bees are quite smart, and they'll know if the color is associated with a change in reward, whether that be pollen or nectar, then they will quickly learn. But in this case, we have yet to detect any differences in nectar or pollen. So the pollinators don't seem to be discriminating between these two. Well, maybe they have slightly different soil chemistries. We did a series of soil collecting. There's Stu from Creekside Science and Don Mao from the Native Plant Society, who generously showed Sharon Strauss and I around, in this case, the Kirby area and the rest of Coyote Ridge to collect soils of the endangered taxa plus its closest relatives. And if we look at things as simple as the calcium-magnesium ratio, which is so distinctive in Serpentine Endemics, we'll see they're well below 1, which makes them serpentine. Yet, whether you are the white-flowered, pink-flowered, purple-flowered taxa, you all have very similar calcium-magnesium ratios. So it doesn't look like the calcium-magnesium is differentiating our taxa. By the way, we threw in an extra site here called the Tulare Hill Reintroduction site that will come up later, but notice that it falls within that range. Just to show you here, if you go up to the top of Mount Hamilton and collect that dark-flowered one, it is quite distinctive in its calcium-magnesium ratio. There's no serpentine atop there, it's shale. Well, we measured 24 soil chemical components. None of them individually came out as distinct for the different taxa, but we put them into a principal components analysis just to confirm, and sure enough, all over the board here, PC1 explaining 33% of the variance, PC2 18%, collectively over half of the variation in those 24 chemical characteristics. And you start to put on the pink ones, and you'll see they're also all over the place, as are the glandulosus taxa that are samples that are mostly purple. And you put the reintroduction site in nicely, it falls in the middle. There are no significant differences in the PC values either. So it doesn't seem like the soil chemistry as measured is distinctive. Maybe the plants can identify something in the soil that we are not measuring. I would give that plants the ability to do that. So we took the plants in, we grew them in a lath house environment in different soil types. We took seeds, for example, from the Metcalf Canyon Jewelflower. We grew them up and measured their dry biomass at maturity, and we put them in 4 different soil types. Their home soil, and then the away soils from their closest relatives, and then conveniently in the reintroduction site. And you'll see, surprisingly, that the least preferred soil, they grow poorest, again, in their home soil. I think this is now becoming a common trend, at least from 3 different taxa in my lab, that when you get to be really rare, your home soil may not be the thing that you are most adapted to. I think we should consider this broadly when we're reconsidering how to take care of endangered species. If you take the stalpy seeds, these are the light pink ones, you'll see that they have a surprisingly similar stair-step pattern. That is, they grow poorest in the soil collected off Metcalf Canyon, intermediate in their own soil, and actually better in their closest relatives in our potential reintroduction site. Just to quickly summarize those results, it was a complicated experiment. We did a lot of replications, and we found that jewelflowers rarely grow best in their own home soil. In fact, the reintroduction site at Tulare Hill harbors plants with the highest fitness, that is in terms of the number of flowers and in terms of the amount of biomass. And lastly, we did find a weak correlation with soil iron and available nitrogen in organic material. That is, in fact, Tulare Hill has a little bit more iron, a little bit more nitrogen in organic material than the other sites. So I'll tell you briefly about the molecules, but this is no silver bullet. Here again is the Streptanthus glandulosus complex, and let me just let you know that there are some recent work on this that has attempted to shed light. A great recent phylogeny by Ivo Cacho and Sharon Strauss et al. That puts together a nice relationship for the jewelflowers, but in fact synonymizes everything within this S. glandulosus complex to this single branch and neglects any differentiation therein. Again, Meyer and Bassetta's work in 2010, I pointed out earlier, has the southern clade, but unfortunately there's only a single sample of albidus in there for them to make any conclusions about. They do draw some conclusions about albidus paramoenus based on only 2 samples, and in fact, I think you can't see the numbers here, but that entire clade is only different in 1 ITS site and 1 chloroplast restriction site. And it's really tenuous to start making conservation statements about 1 or 2 mutations. So we've studied some 624 AFLP markers. These are these hypervariable markers like a DNA fingerprint. They suffer from the exact opposite problem. Instead of not enough variation, we found all but 17 of the 624 markers we studied were variable. And most of the variation exists within populations, whether you are the Metcalf jewelflower, the most beautiful jewelflower, or the others. In fact, a very small amount is actually attributed to among species differences. But we can look at this in a phylogenetic context and see how the taxa fall out. I don't want you to look at the tips here. I just want to let you know that we're looking at some phylogenetic approaches. I've indicated the bootstrap values when they are reasonable, and they are rarely reasonable. So the tree is poorly resolved, but in our best estimate of the tree, all the STALAL samples, except a few, come out together. The stalpy samples come out together, and the stigl samples come out together, and STALAL can be defined as that node, except there are a couple of exceptions. This is STALAL over here, and a STALAL over here, and a couple of stalpies that creep into the STALAL. So we do see some molecular differentiation, but it's not complete. It's definitely not as good as the flower color. Let me just briefly walk you through how we've taken this data into the field. I realized that in submitting my final report and preparing the manuscript that nothing really was going to come of this. And Stu encouraged me to go throw some seeds out there. In fact, we ended up assembling, with the help of Crystal, a somewhat respectable reintroduction plan that someone funded for us through the Bureau of Reclamation. And we took seeds from all over the species range using the Metcalf Motorcycle Park as a control. We attempted to reintroduce the jewelflower at Tulare Hill. In fact, we picked 4 sites using some remote weather data to pick those sites, as well as the amount of sunlight that those are exposed to. That site, by the way, it was known to have jewelflowers in 1980. It was extirpated by development, but probably suffered from the lack of cattle most recently. And we collected seeds from as many populations as we could find. We maintained what the authorities called a captive breeding population. You can see it there, fenced in with a chain, and for fear of falling off of the plates, the pallets that we put them on there, We had over 200 plants in 2013 and some 100 plants in 2014 generating seeds. Those were way too many fruits for us to handle, so thanks to a bunch of generous Native Plant Society volunteers at Santa Clara University working alongside Santa Clara undergraduates and some Creeksiders like Crystal and Leck in particular. We cleaned all those seeds. We ended up with over 80,000 seeds, which were then planted. That's actually March 2014. Hard to believe that we were wearing raincoats. But yeah, that was the one rain. We waited until the one rain. And we did it again in October of 2014 when only a dozen of these plants germinated, and in fact none of them made it to flowering. We thought it was unfortunately a complete failure. About 100 seeds in each site, and as of December 31st of this year, this is New Year's Eve, we had our first Jewelflower sightings. This picture taken by Crystal Nieder. There's one there, and the current count is 4,733 Jewelflowers. Wow. The majority of the plots actually have a Jewelflower in it. This is just the percent of plots, and you'll see, in fact, we get up to 100% of plots with Jewelflowers in them. Whether they were seeded in the rain there, yet didn't come out, or whether they were seeded in the dry fall, you'll see a huge proportion of our several hundred plots with Jewelflowers. They average only 5 or 6 plants per plot, but we expect to see an established population of a couple thousand plants there in the next couple years. With that, I just want to make sure I thank some of the most important contributors here. Aaron's a technician in my lab and has done a huge amount of the legwork. Here's our planting crew. That's Aaron and I and the rest of the Creekside crowd. And Dawn Mail at the CNPS, in particular, shout out to Stu and Crystal. Thanks so much.
If you have any questions, I'd be glad to answer.